Okay, so can you see my slides? Yes?
Perfect.
So very happy to tell you a bit about our work on host microbiome interactions today, mostly on host microbiome interactions, and I'll say just a word about indirect genetic effects as well. So I wanted to present first or remind you of the data set that we have. So the data set is based on the P50, the 5,000 rats from the first P50, for which genotypes and phenotypes are available, thanks to you all. And on a subset, a large subset of those rats, fecal microbiome data were also collected in the form of 16S data. And then on a smaller subset of rats, we also have fecal metabolome data, untargeted, and we also have shallow shotgun sequencing of the fecal microbiome. And hopefully, you know, this data set can be increased, tripled, hopefully, I said by 2024, but you know, that's an idea. And I have in mind that Paul, Suzanne, Tom, and Olivier at least are collecting Zika on the new rats. And so I'd love to sort of have a very quick chat or messages with each one of them to, you know, hear whether this is still happening and so on, and talk about this a bit more. So I wanted to talk about expected publication because I was asked to, and this is, I'm going to tell you a bit of our results as well as we go along. So I guess the first publication that I expect is one focusing on host genetic effects on gut microbiome and metabolome composition, and also effects on host phenotypes. That will be based on 16S data only. And so we'll start with a comparison of the rat microbiome composition with the better known mouse and human microbiomes. We'll briefly discuss important covariates. For example, we've seen that there are strong differences between the phenotyping center, which has been reported before, but we see very little effects of sex, which I think is also consistent with a lot of studies. We'll have results on heritability, and so I'll show you some of these results now. These are three questions that I've identified as potentially most interesting. The first one is the microbiome heritable? So in the literature, we've read the two opposites. We've read in humans that really the environment dominates over host genetics, and that was published in Nature 2018, whereas in wild baboons, a paper recently came out that was showing that the gut microbiome is almost universally heritable. So why this very strong difference? Well, it seems to me that it's just a matter of design, because in humans, you know, they had only one microbiome sample per individual, whereas in baboons, they followed the baboons longitudinally. And so they collected a lot more data, and basically heritability just tracks this. So I want to see what is it like in our data set, and then we can ask, because the rats were, you know, sent to 3 different centers, Memphis, Tennessee, and Michigan. And in Tennessee, we also have breeders. We have microbiome from breeders, and we have microbiome from the behaviorally phenotype rats. We can see whether heritabilities of taxa are correlated across these phenotyping centers and studies. There are 4 in totals. And also we want to compare the microbiome traits to the other phenotypes that are available for this sample. And I think that's quite unique that's what you need to have both microbiome and lots of phenotypes. So, because heritability is quite population specific, it's interesting to compare the traits. So here are the results for the question, is the microbiome heritable? I chose to plot here QQ plots of heritability p-values. So we have the 4 studies I was mentioning before here in New York, Michigan, Tennessee behavior, and Tennessee breeders. And then each, so these are QQ plots, and each dot here is for a microbial taxa. And so if they were not heritable, we would expect the dots to fall along the red line here, but because all the dots, at least in New York, for example, are above the red line, it's not the diagonal because of the scale, but it's above the red line, that is clear indication that taxa, microbial taxa, tend to be heritable. And that's true in New York, in Michigan as well, in Tennessee behavior, not so much in Tennessee breeders. And I'm looking into this. Now, are taxa heritabilities correlated across studies? The short answer is not really. There's little to no evidence of a correlation really, except in that case, which is between New York and Tennessee behavior, where there's a slight correlation, which is actually significant even after von Ferroni correction. But you know, between New York and Michigan, for example, the heritabilities are largely, well, they are uncorrelated. And then something that I think is really interesting, because it puts the microbiome results into context, is to compare with the behavioral and non-behavioral phenotypes. So this is showing the heritability of various traits. Here in very light, we have microbial taxa in Tennessee breeders, then in Michigan, in New York, in Tennessee behavior. Orange are for heritabilities for behavioral phenotypes, and non-behavioral phenotypes are in red. So first of all, you can see that non-behavioral phenotypes tend to have higher heritabilities than behavioral phenotypes. And then the microbiome is even further to the left, meaning that heritability for microbiome traits is generally even lower than it is for the behavior. So that's sort of consistent with, you know, both the human and baboon results really. The traits are heritable, but heritability is low. So you really need an experimental design that can capture heritability and increase heritability by reducing noise to be able to detect this. So now in this first paper, I think we should also have QTLs, of course, and both at the level of the overall microbiome composition, the beta diversity matrix, and at the level of individual taxa. And we'll do a bit of GWAS as well. So I'll show you one result, which is by far the most sort of striking, robust result. And why I'm saying it is robust is because we detect the same QTL, a very strong QTL, in all 4 studies. So we have Michigan, New York. So I actually don't remember which one is which, but these are the 4 studies: Michigan, New York, Tennessee breeders, Tennessee behaviors. In one, so this is a locus zoom plot, a QTL where we've zoomed in. The QTL reaches log P24 in one of the studies, 10 in another, 7.5 in the other, 6 in the other. This is the exact same region. So you can see that this host genetic effect on Paraprevotella abundance is really detected in all 4 studies. And that's really important because it means if we study the same HS rats potentially in a 4th center or 5th center, we probably would detect the same association. So it's also a tractable result if we wanted to dissect it. And that locus is also significantly associated with fasting glucose in a GWAS analysis, and that's after accounting for multiple testing, of course. So this is, you know, the kind of results that I'm going after in this first paper, the most striking results. So at the moment, I'm working on the analysis of the metabolome data. In the past, a very long time ago, when I did a first round of analysis, I found a QTL for many bile acids detected using the metabolome data that was mapping to that gene here, SLC10A2. And I was really excited because the same result has been detected and experimentally validated in diversity outbred mice. And so for me, this is sort of a positive control. And then I improved the data processing pipeline, not so much the genetic analysis, but for example, in untargeted metabolomics data, we can we get a big table with lots of features in columns and rats in rows, and there are many zeros here because of the way the LCMSMS is done. We don't detect the same peaks in all the rats, but we can go back and actually merge some of those peaks and fill fill in the zeros. So this is something that's recommended for LCM-SMS data, and I'm now doing this. But then when I analyze these data, the QTL has vanished. And because I was considering as a sort of positive control, I'm taking a bit of time here to try to see, what's happening, what step is really important here. So second publication that I like to work on, and for which I actually got a small fellowship / grant, is to do a systematic characterization of the causal relationship underlying host microbiome correlation. So we get lots of we get lots of correlations between microbiome traits and phenotypes of the rats. I would like to determine whether the microbiome is influencing the phenotype, whether the phenotype is influencing the microbiome, or whether the two are independent, potentially resulting from a confounding factor. And to do this causal analysis, I want to leverage the genotype of the rats along the principles of Mendelian randomization, for example. But so I want to use several techniques, not all of which have been used in animal models. I think I'm not aware of people using Mendelian randomization in lab mice or rats, so I'd like to hear if anyone is. But so we'll try to implement a few different analyses and see how well we can do here. And then a third publication would make use of the shallow shotgun data that we already have. But I think at the moment we cannot really do much with just the shallow shotgun data. They are really too shallow. And also we found out that most of the reads map to the host, to the rat, as opposed to the microbiome. And that's potentially because the rats were fasted overnight. And so there were fewer bacteria in their guts. So we get lots of host reads, which I'm doing something with, as I'll say in a minute. But so to be able to use the shallow shotgun data we have at the moment, I think we need to get basically an imputation panel by collecting dense shotgun data on a smaller, much smaller subset, on a smaller sample of HS rats. And with that, we could do what is called metagenomes assembled genomes. So we inferring the genomes of that bacteria. We could compare with mice and rats, as was recently done for mice. We could get a catalog of genes that can be used like 16S for microbiome analysis, but there's a value in having more than just one gene to do these analyses. And then we could do a functional profiling of the rat microbiomes and then test for association with the phenotypes. So today I wanted to take this opportunity to sort of ask you whether you'd be interested in helping me collect new data. So that would be for anyone really who's working with rats. I think it should be focused on HS rats, but if we have data from other rats, I think that would be valuable too. And the idea would be to collect this dense shotgun data to make this imputation panel. So we'd collect fresh fecal microbiome samples on a few rats, and few rats can count on the fingers of two hands probably. I'd like to vary the fasting state because I think that influences the number of bacteria present in the gut and also the types of bacteria. I'd like to vary the collection technique, whether we squeeze the cecal content out and then we would have the bacteria that are really in the lumen of the tube, whereas we could open up the cecum and scrape the mucosa to get the bacteria that are more closely associated with the mucosa. And we could perhaps collect fecal material as well. So I'll be staying online during the break, and I'd really like to hear, maybe just drop me a line if you think you might be interested, and then I'll follow up by email. What I would do then is to get the dense shotgun sequences, and then we would be taking care of the analyses here. And so I'll finish with the current project that we're working on. So I just started my lab at the CRG in Barcelona, and I have 2 PhD students who just started 1st of October.
And when you say you'll finish, I think you'll finish relatively quickly because we're sort of at the time now. I know you started a couple minutes late.
Yes, I'm. This is my last slide.
Chapter.
So just wanted to say that we are trying to use the reads that align to the host from the shallow shotgun data to quantify allocoprophagy. And to do this, we're making use of a paper that Carl Broman and team recently published to be able to quantify allocoprophagy from when we detect DNA from the cage mate in the gut of a focal rat. And so this is the part where that should allow me to study indirect genetic effects as well, but I'll skip and conclude by thanking everyone involved here, and of course the consortium, but also the Center for Microbiome Innovation at UCSD.